Students, I am Dr. Sucheta Das Maji, Associate Professor, Department of Biotechnology, Huldia Institute of Technology. Today I am going to discuss one important topic of soil microbiology, biological nitrogen fixation, which is a vital ecological process that plays a vital role in maintaining the balance of nitrogen in ecosystem. What is nitrogen fixation? Fixation of elemental nitrogen in atmosphere by the microorganisms through a reductive process into ammonia is called as biological nitrogen fixation. Nitrogen is an essential element for life. It is a major component of chlorophyll, the most important pigment needed for photosynthesis. Also, it is found in amino acids, the key building blocks of proteins. It is also found in other important biomolecules such as adenosine triphosphate and nucleic acids. Even though nitrogen gas is one of the most abundant elements in Earth's atmosphere, plants can only utilize the reduced form of this element, mainly combined or fixed forms of nitrogen such as ammonia and nitrate. Plants acquire these forms of combined nitrogen by the addition of chemical fertilizer or manure to soil or the release of these compounds during organic matter decomposition or through natural processes like lightning and most importantly through the biological nitrogen fixation. Much of the nitrogen is provided to the cropping systems in the form of industrially produced nitrogen fertilizers. But use of these fertilizers had led to worldwide ecological problems such as the formation of coastal dead zones. Biological nitrogen fixation, on the other hand, offers a natural means of providing nitrogen for plants. It is a critical component of many aquatic as well as terrestrial ecosystems across the biosphere. Biological nitrogen fixation was discovered by scientist Beijering in 1901. This process accounts for about 70% of total nitrogen fixed in the biosphere. A variety of prokaryotic microorganisms have the ability to reduce the atmospheric nitrogen. Based on the growth pattern, the nitrogen fixing microorganisms or nitrogen fixers can be divided into three groups: the free living nitrogen fixers, associative nitrogen fixers, and symbiotic nitrogen fixers. Many heterotrophic bacteria live in soil and fix significant levels of nitrogen without the direct interactions with other organisms. Examples of this type of nitrogen fixing bacteria include species of Azotobacter, Bacillus, Clostridium, and Klebsiella. These organisms get energy for nitrogen fixation by oxidizing organic molecules released by other organisms or from decomposition. There are some free living organisms also. They have the ability to use the inorganic compounds as a source of energy. As the nitrogen fixing process can be inhibited by oxygen, free living microorganisms behave as anaerobes or microaerophiles while fixing the nitrogen. Due to the scarcity of suitable carbon and energy sources for these group of microorganisms, their contribution to the global nitrogen fixation rates is generally considered minor. Associative nitrogen fixers occur on the surface of the plant, mostly in the root zone. The nitrogen fixers remain over the surface of the plant root, and these type of nitrogen fixers can be found in the rhizoplane as well as in the rhizosphere. This process is carried out by representatives of the genera Azospirillum, some of the Azotobacter, and Acidobacter. These bacteria contribute to nitrogen accumulation by tropical grasses. Recent evidence suggests that their major contribution may not be in nitrogen fixation but in production of growth promoting hormones that increase the root hair development and thus greater ability of the plant to take up the nutrients. Most important nitrogen fixers are the symbiotic nitrogen fixers. Many microorganisms fix nitrogen symbiotically by partnering with a host plant. The plant provides sugars from photosynthesis that are utilized by nitrogen fixing microorganisms for the energy it needs for nitrogen fixation. In exchange for these carbon sources, the microbe provides fixed nitrogen to the host plant for its growth. By far, the most important nitrogen fixing symbiotic associations are the relationships between legumes and rhizobium and bradyrhizobium bacteria. Important legumes used in agricultural system include alfalfa, beans, clover, cowpeas, lupins, peanut, soybean, etc. Another important example of this group of nitrogen fixation is with the water fern azolla uh, with cyanobacterium Anavina azuli. Anavina colonizes cavities formed at the base of the azolla fronds. There, the cyanobacteria fix significant amounts of nitrogen in specialized cells called heterocysts. This symbiosis has been used for at least 1,000 years as a biofertilizer in the wetland paddies in Southeast Asia. Rice paddies are typically covered with azolla blooms that fix up to 600 kg nitrogen per hectare per year during the growing season. In this slide, we can see the basic nitrogen cycle, and one important part of it is biological nitrogen fixation. Some other important steps which are involved in the movement of nitrogen in the biogeochemical cycle are nitrification, nitrogen assimilation, denitrification, and Anammox reaction. Among these reactions, some are aerobic and some are anaerobic in nature, and some can happen both in aerobic and anaerobic environment. Nitrification is the aerobic process of ammonium ion oxidation to nitrite and subsequent nitrite oxidation to nitrate. Bacteria of the genera Nitrosomonas and Nitrosococcus, for example, play important roles in the first step, and Nitrobacter and related chemolithoautotropic bacteria carry out the second step. Nitrogen assimilation occurs when inorganic nitrogen is used as a nutrient and incorporated into new microbial biomass. Ammonium ion, as it is already reduced, can be directly incorporated without major energy costs. The primary route of ammonia assimilation is the formation of amino acids. Some microorganisms form the amino acid alanine in a reductive amination reaction catalyzed by alanine dehydrogenase. Again, many bacteria and fungi employ glutamate dehydrogenase at least when the ammonia concentration is high. This is the major route of ammonia assimilation. A second route of ammonia incorporation involves two enzymes acting in sequence, glutamine synthetase and glutamate synthase. This route is present in E. coli, Bacillus megaterium, and other bacteria. The two enzymes acting in sequence operate very effectively at low ammonia concentrations, unlike the glutamate dehydrogenase pathway. A significant amount of energy expenditure is associated with assimilatory nitrate reduction. Nitrate first be reduced to ammonia before the nitrogen can be converted to an organic form. This reduction of nitrate is called assimilatory nitrate reduction. This process is widespread among bacteria, fungi, and algae. Assimilatory nitrate reduction takes place in the cytoplasm of bacteria. The first step in nitrate assimilation is its reduction to nitrite by nitrate reductase enzyme, and nitrite is next reduced to ammonia with a series of 2 electron additions catalyzed by the same enzyme nitrite reductase, where nitroxyl and hydroxylamine are originated as intermediate. Ammonia can be oxidized under anoxic conditions by the bacterium Brocadia in the process called anamox. In this reaction, ammonia is oxidized anaerobically with nitrite as the electron acceptor, forming nitrogen as the final product, which is released to the atmosphere. Although a major process in sewage and anoxic marine basins and sediments, anamox is not significant in well-drained soils. The process of denitrification, which is a dissimilatory process, nitrate is used as an oxidant in anaerobic respiration and usually involves heterotrophs such as Pseudomonas denitrificans. The major products of denitrification includes nitrogen gas and nitrous oxide. Sometimes nitrous oxide can photochemically oxidize to nitric oxide in the atmosphere, which reacts with ozone to produce nitrite. Nitrite is of environmental concern because it helps in formation of carcinogenic nitrosamines. Finally, nitrate can be transformed to ammonia in dissimilatory reduction by a variety of bacteria including Geobacter species, Desulfovibrio species, and Clostridium species. So, the reduction of atmospheric gaseous nitrogen to ammonia is called nitrogen fixation. This reduction process is catalyzed by the enzyme nitrogenase. Nitrogenase is a complex system consisting of two major protein components, a molybdenum iron protein whose molecular weight is around 220 kd, also called nitrogenase, joined with one or two iron proteins whose molecular weight is around 64 kd and also called the nitrogenase reductase. The molybdenum iron protein contains 2 atoms of molybdenum and 28 to 32 atoms of iron. The iron protein has 4 iron atoms. Though the enzyme bound intermediates in this process are still unknown, it is believed that nitrogen is reduced by 2 electron additions. The reduction of molecular nitrogen to ammonia is quite exergonic, but the reaction has a high activation energy because molecular nitrogen is an unreactive gas with a triple bond between the two nitrogen atoms. Therefore, nitrogen reduction is expensive and requires a large ATP expenditure. At least 8 electrons and 16 ATP molecules, means 4 ATPs per pair of electrons, are required. The electrons come from ferredoxin or flavodoxin that has been reduced in a variety of ways by photosynthesis in cyanobacteria, respiratory processes in aerobic nitrogen fixers, or fermentations in anaerobic bacteria. For example, Clostridium pasteurianum, the one anaerobic bacterium, reduces ferredoxin during pyruvate oxidation, whereas the aerobic azotobacter uses electrons from NADPH to reduce ferredoxin. If we study the electron flow in details, we can find that iron protein is first reduced by ferredoxin, then it binds ATP. ATP binding changes the conformation of the iron protein and lowers its reduction potential, enabling it to reduce the molybdenum iron protein. ATP is hydrolyzed when this electron transfer occurs. Finally, reduced molybdenum iron protein donates electrons to atomic nitrogen. The reduction of nitrogen to ammonia occurs in 3 steps, each of which require an electron pair. 6 electron transfer takes place, and this requires a total 12 numbers of ATPs per nitrogen reduced. The overall process actually requires at least 8 electrons and 16 ATPs, because nitrogenase also reduces protons to hydrogen. The hydrogen reacts with diamine to form nitrogen and hydrogen. This futile cycle produces some nitrogen even under favorable conditions and makes nitrogen fixation even more expensive. Symbiotic nitrogen fixing bacteria can consume almost 20% of the ATP produced by the host plant. Nitrogenase can reduce a variety of molecules containing triple bonds like acetylene, cyanide, and azide. The rate of reduction of acetylene to ethylene is used to estimate nitrogenase activity. Once molecular nitrogen has been reduced to ammonia, the ammonia can be incorporated into organic compounds. In the symbiotic nitrogen fixer rhizobium, it appears that ammonia diffuses out of the bacterial cell and is assimilated in the surrounding legume cell. Nitrogenase is quite sensitive to oxygen and must be protected from oxygen inactivation within the cell. In many cyanobacteria, this protection against oxygen is provided by a special structure called the heterocyst. Sometimes the presence of leg hemoglobin also helps in the protection of the nitrogenase complex from aerobic destruction. Now I want to discuss briefly the rhizobium legume symbiosis process and how this relationship between a bacteria and a plant is established specifically. The rhizobium infection process is controlled by the BACA gene that is required to establish the nodule. Symbiotic development in legumes is controlled by a plant regulator protein that differentiates root cortical cells and restarts cell division to establish nodule primordia within the invaded root. This complex infection process involved a series of molecules produced by the host plant that led to the exchange of recognition signals. Flavonoid inducers produced by the plant play uh, a major role in this process by stimulating the rhizobium to synthesize specific node factors that activate the host symbiotic processes necessary for root hair infection and nodule development. After bacterial attachment, the root hair curl and the bacteria induce the plant to form an infection thread that grows down the root hair. The rhizobium then spreads within the infection thread into the underlying root cells. Within a few minutes, the rhizobium actually enters the plant cytoplasm while it is in the infection thread. When the bacteria released from the infection thread into the host cell, the rhizobium is enclosed by a plant derived membrane called the peribacteroid membrane to form a bacteroid. Further growth and differentiation led to the development of a nitrogen fixing form, a structure called a symbiosome. One specific nodule components called the leg hemoglobin protect the nitrogen fixing enzyme from oxygen and they are produced to complete the nodulation process. The symbiosomes within the mature root nodules are the sites of nitrogen fixation. Within these nodules, the nitrogen is taken up by the bacteroid and form the ammonia and then subsequently alanine and other amino acids and then distributed throughout the plant. And the symbiosome form lack the production ability and cannot revert back to the bacteria again. In the first picture, we can see that plant root releases flavonoid that stimulate the production of various node metabolites by rhizobium. There are many different node factors that control infection specificity. In the second picture, attachment of rhizobium to root hairs involves specific bacterial proteins called ricadhesin and the host plant lectins that affect the pattern of the attachment and node gene expression. In third, it is a structure of a typical node factor that promotes the root hair curling and plant cortical cell division. In the fourth picture, we can see that a plant root hair covered with rhizobium and undergoing curling. In the fifth, initiation of bacterial penetration into the root hair cell and infection thread growth, and it is coordinated by the plant nucleus, which is denoted as capital N. Then uh, a branched infection thread shown in electron micrograph. Then cell to cell spread of rhizobium through transcellular infection threads, followed by release of rhizobia and infection of host cell. Then formation of bacteroids surrounded by plant-derived peribacteroid membranes and differentiation of bacteroids into nitrogen-fixing symbiozone. Here we can see that the bacteria changes morphologically totally and enlarged around 7 to 10 times in volume. The symbiosome contains the nitrogen fixing bacteroid, a peribacteroid space, and the peribacteroid membrane.